Hello and welcome, students. Today, we are about to embark on a practical that brings together several key concepts we've explored in our study of ecology. So far, we've delved into the dynamic interactions within ecosystems. We've explored how flora and fauna compete for resources and the delicate balance of interdependence among species. These foundational topics have given us a good sense of how living things interact with each other and their environment. This practical leads us further into applied ecology, as we investigate how to measure the population size of a common species in a habitat. We will use different sampling techniques to examine how various factors influence the distribution of this species. Our goal is to see firsthand how the competition for resources and the interdependence we've talked about affect real-world ecosystems. By quantifying population sizes and examining the effects of environmental factors, you will gain practical insights into the ecological concepts we've discussed. Let's get started. Grab your equipment and let's explore the living world around us. Before we dive into the details of our practical, let's take a moment to appreciate why this activity is so valuable. Firstly, understanding how to measure the population size of a species in a habitat and how environmental factors influence their distribution is directly applicable to real-world ecological and environmental management scenarios. For example, conservationists use these techniques to monitor endangered species and assess the impact of habitat changes. Secondly, this practical equips you with essential skills in data collection and analysis through sampling techniques like quadrats and transects. These are foundational methods used in professional ecological research. By practicing these techniques, you gain experience in designing fair tests, collecting unbiased data, and interpreting results accurately. In summary, this practical isn't just about learning a set of techniques. It's an opportunity to apply your biological knowledge to real-world issues, all whilst developing critical scientific skills and enhancing your problem-solving abilities. Let's get started and see how environmental factors can influence the living organisms around us. Let's prepare our practical. We begin in our designated outdoor study area, which we will divide into a grid using tape measures and stakes. We will study the shaded area. This systematic approach will help ensure that our sampling is rigorous and unbiased. The study area is subdivided into a grid of equal sections. This is done by measuring out a series of lines that cross the area, forming a checkerboard pattern on the ground. We'll then use a random number generator to determine the coordinates for quadrat placement within the grid. This randomness is crucial for an unbiased sample. Next, we need to position the quadrat. At each set of coordinates, we will carefully place a 1 meter squared quadrat frame on the ground. Within the quadrat, every individual organism of the target species, such as daisies, will be counted. This step is repeated multiple times at different coordinates to ensure our sampling is thorough. Once all quadrats have been sampled, we calculate the mean number of organisms per quadrat in our designated area. Using our data, we found 23 daisies in total over 10 quadrats. 23 divided by 10 equals 2.3. So, on average, there are 2 daisies per meter squared in that area. The entire process is then repeated in a second sample area that represents a different environmental condition. For example, if we just sampled a shaded area moved to an open area, you might find an average of 2 daisies per meter squared in shaded areas, and 22 daisies per meter squared in open fields. This comparison helps us understand how the distribution of the species varies with changing factors. Next, we will use a different measuring device, a transect, to collect information systematically about population abundance across an environmental gradient. Note that this is not a random sampling method, as it samples species number at set intervals along the transect. To set up the transect, we mark a straight line through a different part of the habitat using a long tape measure. This will act as our transect line. For example, you may set the transect line up a hillside to investigate the effect of altitude on daisies. Along this transect line, quadrats are placed at regular intervals, such as every 2 meters. This systematic spacing helps us capture gradient changes in environmental factors. We will then record the number and type of organisms present within each quadrat along the transect, as well as the abiotic factor at each quadrat, such as altitude. For vegetation like grass, we might estimate the percentage cover instead of counting individual blades. All data gathered, including counts and percentage covers, are then carefully noted in our field logs. These will be used later for analysis. After collecting our data, we ensure all equipment and materials are cleaned and stored appropriately. Now let's take a look at an overview of the biological concepts relevant to this practical. Organisms live in different places due to varying environmental conditions. The specific location where an organism lives is called its habitat. The distribution of an organism refers to where it is found within its habitat, and this distribution is influenced by environmental factors. For example, daisies might be more abundant in open areas with more light compared to shaded areas under trees. Let's also look at the key steps and their importance. Firstly, we used quadrats. This is important as quadrats provide a standardized way to measure organism abundance and distribution. Secondly, we used transects. Transects offer a method to study how organism distribution changes across an environmental gradient, such as level of light or moisture in the soil. Now let's model and analyze the data collected from quadrats and transects. First, we need to calculate the mean abundance. For quadrat data, calculate the average number of organisms found per quadrat to get a sense of species abundance in different areas. Using our data, on average, there were 2 daisies per meter squared in that area. When you repeat the experiment in a different environment, you may get different results. For example, you might find an average of 2 daisies per meter squared in shaded areas and 22 daisies per meter squared in open fields. Next, we need to compare the mean values. To do this, compare the means between different habitats or areas to understand how environmental factors influence species distribution. For example, if light intensity is the varying factor, higher mean values in illuminated areas indicate that light positively affects daisy distribution. Next, we estimate the population in a survey area. To achieve this, we can use this equation. Estimated population size equals total area divided by area sampled, multiplied by total number of organisms counted. For example, if the total survey area is 100 meters squared, and we sampled 10 meters squared by placing 10 1 meter by 1 meter quadrats, finding a total of 23 daisies, we would calculate the estimate using this equation. 100 divided by 10 multiplied by 23 equals 230. Next, we need to create graphs to visualize the data. Plotting the mean abundance against different environmental conditions can help identify patterns and relationships. This visual representation aids in understanding the influence of certain factors on the distribution of species. In our graph, we can see that as the altitude increases, the number of daisies decreases. Then, discuss the findings in the context of environmental factors influencing organisms. For instance, more daisies in open fields suggest that light is a crucial factor for their growth. Conversely, higher moisture levels in another area could explain the increased abundance of a different plant species. You should also consider other possible influencing factors like soil composition, nutrient availability, or human activity that might affect the results. Finally, consider any anomalies. If some quadrats or transect points show anomalous results, which are unusually high or low counts, consider possible causes such as experimental error, unique microhabitats, or disturbances in those particular areas. In today's lesson, we conducted a practical experiment to measure the population size of a common species in a habitat and investigated how a specific factor influences its distribution. The key steps of this practical were quadrat sampling, in which we placed quadrats randomly within our study area to obtain sample counts of the species present. Transect sampling, in which we used transects to systematically sample species across an environmental gradient. Then, we estimated population size using the data from our quadrats. Next, we selected a specific environmental factor, such as light or moisture, and noted its value at each quadrat position. Finally, we compared the abundance of our species with the varying levels of the chosen factor to observe patterns in distribution. Our key takeaways for future practicals are to ensure random and unbiased placement of quadrats to get representative samples, and to be consistent in measuring and recording environmental factors to maintain data reliability. This practical not only enhanced our skills in ecological sampling and data analysis, but also deepened our understanding of how environmental factors affect species distribution within ecosystems.